Imagine you're a detective, right, tasked with finding a murderer who has completely dismantled a house.
Okay, I like where this is going.
So our detective, let's call him Medlock Holmes, he walks up to the address, and he's expecting to see a crater, a completely destroyed property.
Right.
But the house is standing, the roof is totally intact, the foundation is solid. It's really only when he walks inside that he realizes something is, well, profoundly wrong.
The structure is there, but the function is gone.
Exactly. The house hasn't been destroyed. It has been fundamentally disconnected. Like the doors between the rooms have just been removed. The phone lines to the outside world have been severed.
Oh, wow.
Yeah, and the thermostat has been wired to the oven, pushing the heat to a boiling point in the kitchen while the bedrooms are freezing.
That is a terrifying environment to live in.
Right. To the casual observer on the street, it's a perfectly normal house, but for the person trying to actually live inside it, it is chaotic, fragmented, and terrifying. And that is precisely what a psychiatrist is looking at when they examine a brain affected by schizophrenia.
It really is. I mean, it completely upends our standard medical expectations.
How so?
Well, in most of medicine, we rely on the comfort of visible pathology. A fractured femur presents as this jagged white line on an X-ray.
Right, it's obvious.
Exactly. A massive middle cerebral artery stroke leaves a gaping necrotic void on an MRI scan. We crave that binary simplicity. But schizophrenia denies us that comfort entirely.
It's the ultimate manifestation of diagnostic muddy waters.
It really is.
Yeah.
Primarily because psychiatry itself is not just the study of biological tissue. It's the study of how brain, mind, and experience interact. And, crucially, how that breathtakingly complex triadic process can quietly go off course over decades.
Which is really the fundamental philosophy anchoring our deep dive today. We are serving as your conceptual companion to a very dense, definitive text for anyone in the field.
Chapter 097 of Kaplan and Sayok's comprehensive textbook of psychiatry.
Spot on. The chapter is titled Cellular and Molecular Neuropathology of Schizophrenia. And for the clinicians, the psychiatry trainees and the registrars listening, I want to establish our parameters right away.
Yeah, we need to set the ground rules.
Exactly. We're not here to just read a list of proteins at you. Rote memorization of acronyms will not help you when you're sitting across from a patient in acute psychosis.
No, it absolutely won't.
Our objective here is to build a robust, dynamic mental model. We need to understand how the brightest minds in psychiatry actually conceptualize this illness, not as a single smoking gun lesion, but as a cascading failure of interconnected systems.
And to build that model, we really have to start where the postmortem researchers started over a century ago. We have to trace their exact, often frustrating path.
Where do we begin?
We begin at the macroscopic level, holding the whole brain in our hands, looking for that missing house.
The early anatomists must have been so confused.
Oh, they were utterly confounded. Because, grossly, the brain of a patient with severe chronic schizophrenia looks entirely normal.
No obvious damage.
None. There are no Alzheimer's-like plaques or tangles, no Lewy bodies, no gross atrophy. But that macroscopic illusion begins to fracture when we subject the data to massive modern meta-analyses.
Right, because when you weigh thousands of these post-mortem brains and compare them to healthy controls, a subtle signal finally cuts through the noise.
What did they find?
There is a small but highly statistically significant reduction in overall brain weight. And what strikes me about this finding is the correlation.
The age of onset, right?
Yeah. That reduction in brain weight inversely correlates with the age of onset.
Meaning the earlier the developmental trajectory goes off course, the more pronounced the final physical deficit is.
Exactly. And we can actually see the physical consequences of this slight volume loss in living patients, which brings us to a watershed moment in the history of biological psychiatry.
The 1976 Eve Johnstone and Timothy Crow study.
Yes. They published this pivotal study using early CT scanning technology.
And they observed that patients with chronic schizophrenia had ventricular dilatation, specifically an enlargement of the third and lateral ventricles.
Now, I want to pause on the mechanics of that because it's a crucial clinical distinction. The ventricles are the fluid-filled cavities in the center of the brain.
Right.
When we say they are enlarged, we aren't saying the disease is actively expanding the ventricles like a balloon being inflated.
No, definitely not.
We are talking about ex vacuo ventricular enlargement. As the surrounding brain tissue slightly decreases in volume, the ventricles simply passively expand to fill the negative space inside the rigid skull.
And that passive expansion has been replicated across decades of structural MRI studies. But here's the critical trap for a junior clinician.
Go on.
If you order a scan on a 20-year-old patient presenting with a first episode of psychosis, and the radiologist notes enlarged lateral ventricles,
You can use that for a diagnosis, can you?
you absolutely cannot use that as a diagnostic confirmation of schizophrenia. Ventricular enlargement is profoundly nonspecific.
You see it in normal aging, right?
Normal aging, hydrocephalus, chronic alcohol use, and in severe bipolar disorder. Furthermore, high-risk family studies show us something really fascinating.
What's that?
You can find significantly enlarged ventricular volumes in the perfectly healthy, unaffected siblings of people with schizophrenia.
Wow. Which fundamentally alters how we interpret the scan. It isn't a picture of the disease itself.
No, it is a macroscopic marker of genetic vulnerability.
It's a structural echo of shared risk alleles. So the gross anatomy points us to a subtle loss of tissue, but it refuses to give us a definitive diagnosis.
Exactly. To find the real pathology, we have to pull out the microscope.
We have to look at the prefrontal cortex, the PFC, because structural imaging tells us there is a 3 to 12% reduction in gray matter volume there.
And this is where the field made a very logical but ultimately incorrect assumption for years.
The neurodegeneration hypothesis.
Precisely. The prevailing hypothesis was simple neurodegeneration. If you have less gray matter volume, it stands to reason that the disease acts like a lumberjack, actively killing and clearing out neurons in the frontal lobes.
Right, like deforestation. And early postmortem studies using older two-dimensional stereologic approaches actually seemed to confirm this.
They did. They looked at thin, flat slices of the cortex, counted the cells, and reported neuronal loss.
But the chapter spends a significant amount of time highlighting how science self-corrects through methodology.
Yeah, those 2D slices were subject to massive counting biases.
Because it depends on how the tissue was cut and how the cells happen to be oriented, right?
Exactly. When modern neuropathologists introduced unbiased, three-dimensional stereologic approaches techniques like the optical fractionator, which meticulously maps and counts cells within a true 3D spatial volume, the neurodegeneration hypothesis collapsed entirely.
It just completely fell apart.
The 3D stereology revealed a stunning truth. There is absolutely no change in the total number of neurons in the prefrontal cortex of patients with schizophrenia.
Wait, no change at all.
None. The total neuronal count is beautifully, perfectly preserved.
But we have to reconcile this mathematically. If the physical volume of the prefrontal cortex is up to 12% smaller, but the absolute number of neuronal cell bodies is exactly the same.
It defies basic spatial logic unless something else is happening.
Right. So the only mathematical solution is that neuronal density has actually increased.
The cells are packed much more tightly together.
To return to your opening metaphor of the house, it is as if the structural framing of the rooms remains entirely intact, but the hallways, the ductwork, and all the sprawling electrical cables running between the rooms have vanished.
And because all of that infrastructure takes up physical space, its disappearance causes the entire structure to condense.
In neurobiology, we call this the reduced neuropil hypothesis.
The neuropil. It's a term we throw around a lot, but let's actually define it tangibly.
Good idea. We aren't talking about the cell bodies, the somas.
No. We are talking about the dense, virtually impenetrable forest of axonal arbors sending signals, dendritic trees receiving signals, and the trillions of synaptic spines bridging the gaps.
The gray matter is a composite of neuronal cell bodies, glial support cells, and this massive web of neuropil.
So if the cells are still there, the shrinkage has to be driven by a catastrophic loss of these connections.
And the researchers didn't just find a generalized thinning of the neuropil, did they?
No, they mapped it to a highly specific cortical address.
The most robust, consistently replicated finding is a dramatic reduction in dendritic spine density, specifically on the pyramidal neurons residing in layer 3 of the dorsolateral prefrontal cortex.
Let's unpack the functional anatomy of layer 3 because it explains the clinical presentation so perfectly.
The cerebral cortex is organized into 6 distinct layers. Layer 3 isn't just a random cross-section.
No, it is the ultimate integration hub. Layer 3 pyramidal neurons are responsible for forming the vast cortical networks.
They're the ones communicating across regions.
Exactly. They receive colossal amounts of data from the contralateral hemisphere, from other association cortices, and heavily from the medial dorsal nucleus of the thalamus.
They are the neurons that allow you to hold a complex thought in your mind, compare it to a past memory, and plan a future action.
They are the biological substrate of working memory and executive function.
So if we look at the dendritic spines, these microscopic mushroom-shaped protrusions that act as the physical receiving antennas for chemical signals,
Right.
and we see that layer 3 neurons have been stripped of these antennas, the clinical picture snaps into focus.
It really does. The profound cognitive deficits, the disorganization of thought, the inability to plan. It is the direct behavioral manifestation of a layer 3 integration hub that has literally lost its physical ability to receive signals.
It makes total sense, but I have to play the role of the skeptic here because this is post-mortem tissue.
I know where you're going with this.
These patients lived with a severe psychiatric illness for decades. More importantly, they likely consumed heavy doses of dopamine-blocking antipsychotic medications for decades.
Yes.
How can we possibly be certain that this loss of dendritic spines isn't just a long-term toxic side effect of the medication? What if haloperidol is the lumberjack?
It is a brilliant question, and it is the single most important confounding variable in psychiatric neuropathology.
How do we get around it?
To isolate the effect of the disease from the effect of the treatment, researchers had to design meticulous controlled studies using non-human primates.
Macaque monkeys, right.
Yes.
Yeah.
They took cohorts of macaque monkeys and exposed them to chronic, long-term administration of typical and atypical antipsychotics, perfectly mimicking the human clinical exposure.
And then examine their cortices using the exact same 3D stereological techniques.
Precise.
And the macaques showed no loss of neuropil.
None whatsoever. The chronic antipsychotic exposure did not alter cortical neuronal density, nor did it cause the specific layer 3 dendritic spine loss.
Wow, so that gives us immense confidence that the missing bridges in the prefrontal cortex are an intrinsic pathological feature of schizophrenia itself.
Absolutely. However, the macaque studies did reveal a different vital clue about the medication.
Oh!
While the neurons were unaffected, the density of glial cells, the astrocytes and microglia that support and modulate the neurons, actually increased by as much as 33% in certain cortical layers in the treated animals.
That is fascinating. It suggests that while the medication isn't destroying the neuronal architecture, it is triggering a massive active immune or metabolic response in the background support network.
Exactly. And we will absolutely return to those glial cells when we tackle the inflammation hypothesis later.
So Medlock Holmes has found his missing bridges in the frontal lobe. But schizophrenia disrupts memory, emotion, and sensory filtering as well.
Which means we have to look deeper into the medial temporal lobe, specifically the hippocampus.
The hippocampus presents a deeply paradoxical landscape. We see the same subtle macroscopic markers, right?
Yes, structural MRI confirms roughly a 4% volume reduction, often more pronounced on the left side, and this reduction is present at the very onset of the first psychotic break.
So it's not just a product of chronic illness.
Exactly. And the volume loss isn't uniform. There is a distinct anteroposterior gradient, meaning the anterior portions of the hippocampus bear the brunt of the volumetric decline.
And again, 3D stereology tells us that despite the volume loss, the absolute number of hippocampal neurons remains stable, though the pyramidal cell bodies themselves might be physically smaller.
So we assume the same mechanism as the prefrontal cortex, a loss of neuropil, a thinning of the dendritic trees.
Right. And indeed, in the subiculum of the hippocampus, spine density is severely down. But Kaplan and Sadok highlight an incredible anomaly in table 12.6-1.
The CA3 paradox.
Yes. There is one specific hippocampal address where the rules reverse.
The CA3 region. When neuropathologists used Goldie impregnation, which is a classic staining technique that allows you to visualize the entire dendritic arbor of a single neuron, they found something startling.
What did they find?
On the apical dendrites of the pyramidal cells in the CA3 region, dendritic spine density was not reduced. It was significantly increased.
The brain is actively building more bridges in CA3 while the rest of the temporal and frontal lobes are collapsing. Why would it do that? It feels like throwing extra life preservers onto a specific deck of a sinking ship.
That's a great way to put it. It is a stunning visual representation of network compensation.
Because CA3 is about pattern completion, right?
Exactly. The CA3 region is critically involved in pattern completion, the ability to recall an entire memory from a partial cue.
So if the surrounding hippocampal and cortical networks are failing, the CA3 region may be driven into a state of hyperactive overdrive,
frantically upregulating its synaptic connections in a desperate attempt to maintain information flow.
It completely shatters the idea that schizophrenia is a generalized passive wilting of the brain.
Yes, It is a highly dynamic, subregion-specific rewiring process. Some circuits fail, and others hypercompensate to the point of toxicity.
Which forces us to look at the master switchboard that connects all these failing and overactive regions. The thalamus.
Every sensory input, except olfaction, has to pass through the thalamus before it reaches the cortex.
And the thalamus is central to the theoretical model. We are particularly interested in the mediodorsal, or MDL, nucleus.
Because it has massive reciprocal closed-loop projections with the exact layer 3 prefrontal regions we were just discussing.
Right. If the prefrontal cortex is the executive, the MDL is the chief of staff feeding it information.
Now, if you read the literature from the 1990s, you'd find prominent papers claiming that the thalamus actually was losing neurons.
Yeah, they reported significant decreases in total neuronal counts in the parvocellular and densocellular subregions of the MDL.
It was a very neat, tidy theory. The thalamus dies, so the prefrontal cortex loses its input, leading to cognitive collapse.
It was almost too elegant.
It was.
Yeah.
As the field pushed forward, employing larger sample sizes from broader brain banks and utilizing much more rigorous, unbiased stereological counting methods, those early findings evaporated.
The current consensus, firmly stated in the textbook, is that there is no robust evidence for neuronal loss in the thalamus.
The chief of staff isn't dead.
But the communication is still broken. If the cells in the prefrontal cortex are intact and the cells in the thalamus are intact, but the physical wiring between them is thinning, then the mystery shifts.
From the physical structures to the chemical messages trying to cross the void.
The bridges are gone, so what is happening to the cargo? This brings us to the chamber of neurotransmission.
And for nearly half a century, this chamber was ruled by an absolute monarchy.
The dopamine hypothesis.
Yes. It is arguably the most famous theory in biological psychiatry, and it was born entirely from reverse engineering pharmacology.
Right. In the 1950s and 60s, clinicians observed that drugs like chlorpromazine and haloperidol had a miraculous ability to halt acute psychosis.
Silencing hallucinations and dissolving delusions.
And through subsequent binding assays, researchers discovered that the clinical efficacy of these drugs correlated almost perfectly with their ability to block one specific target.
The dopamine D2 receptor.
The logic chain seemed unbreakable, didn't it? If blocking dopamine stops the symptoms of schizophrenia, then the root cause of schizophrenia must be an excess of dopamine, hyperdopaminergia.
But the clinical reality immediately began to poke holes in this logic chain.
Because it didn't cure everything.
Exactly. Blocking D2 receptors worked wonders for the positive symptoms, the hallucinations and paranoia. But it did absolutely nothing for the negative symptoms, like blunted affect or avolition.
And it certainly didn't reverse the cognitive deficits. In some cases, it actually worsened them.
In fact, studies mapping the prefrontal cortex showed that normal working memory requires a highly specific Goldilocks level of dopamine.
Too much is bad, but too little is disastrous.
Yes. Non-human primate studies demonstrated that if you chemically deplete dopamine specifically in the prefrontal cortex, you induce profound cognitive deficits that perfectly mimic the negative and cognitive symptoms of schizophrenia.
This forced the field to evolve from a simple excess model to a regional imbalance model.
Hyperactivity of dopamine in the subcortical striatal regions driving the psychosis, coupled with a simultaneous crippling hypoactivity of dopamine in the prefrontal cortex driving the cognitive decline.
That imbalance model is elegant. But this is a chapter on postmortem molecular neuropathology. When researchers finally had the tools to slice into the postmortem brain and actually measure the dopamine receptors looking for the physical scar of this imbalance, what did they find?
They found a ghost.
A ghost?
The evidence for primary structural dopamine pathology in the postmortem brain is, to be blunt, incredibly weak. Let's look at the receptor families.
Okay, the D1 receptor, which is heavily concentrated in the prefrontal cortex.
Its expression is largely unchanged, or findings are wildly inconsistent.
What about the famous D2 receptor? Surely there are too many D2 receptors, which is why the subcortical regions are hyperactive.
They did find elevated levels of D2 receptors in the striatum.
Yep.
But, and this is a massive, but almost all of these brains came from patients who had taken D2 blocking antipsychotics for decades.
Ah, receptor kinetics.
Exactly. Basic receptor kinetics dictate that if you pharmacologically blockade a receptor system, the neuron will upregulate its expression, building more receptors to try and capture whatever neurotransmitter it can.
So what happened when they looked at unmedicated brains?
When researchers looked at the rare postmortem brains of patients who had never been medicated, the D2 receptor density was normal.
Wow. The D2 increase wasn't the disease, it was the biological scar of the treatment.
Exactly.
Okay, so D1 is a bust, D2 is an artifact. I remember in the 1990s there was massive hype around the D4 receptor.
Oh, the D4 hype was monumental.
Because clozapine, the most effective medication we have for treatment-resistant schizophrenia, has a uniquely high binding affinity for the D4 receptor.
But again, molecular reality set in. Neither genome-wide association studies nor definitive postmortem protein assays could conclusively link D4 alterations to the underlying pathology.
And pharmaceutical companies poured millions into developing highly selective D4 receptor antagonists.
And in clinical trials, they completely failed to treat psychosis.
Even the dopamine active transporter, DB, the vacuum cleaner that clears dopamine out of the synapse, is fundamentally unaltered in postmortem studies.
It is a stunning collapse of the theory. The postmortem brain essentially rejects the idea that a broken dopamine receptor is the root cause of schizophrenia.
But we cannot discard dopamine because living patients tell a completely different story.
This is where we must bridge postmortem static anatomy with in vivo dynamic imaging.
Right, PET scans.
Yes. While the receptors might look structurally normal after death, PET scans of living, unmedicated patients experiencing acute psychosis show definitive, profound dopamine dysregulation.
Specifically, they look at the uptake of radiolabeled LDOPA to measure the brain's capacity to synthesize dopamine.
Yes, nascent dopamine synthesis. In living patients, the presynaptic capacity to synthesize and release dopamine in the associative striatum is massively increased.
And when you challenge these patients with a mild amphetamine, their brains dump significantly more dopamine into the synapse compared to healthy controls.
And the volume of that dopamine dump correlates almost perfectly with the severity of their psychotic symptoms.
So we are left with a vital clinical distinction here. Dopamine is absolutely the final common pathway driving the manifestation of psychosis.
But the normal postmortem receptor data tells us that the dopamine system itself isn't structurally broken. It is being driven crazy by something upstream.
The dopamine system is a healthy engine being redlined by a malfunctioning accelerator pedal.
Which logically demands that we investigate the primary accelerator pedal of the brain.
Glutamate.
The excitatory engine.
Glutamate drives nearly 80% of the brain's synapses. And just as the dopamine hypothesis was born from observing how drugs cured the disease, the glutamate hypothesis was born from observing how drugs mimic the disease.
The ketamine clue. It is arguably the most powerful pharmacological model of schizophrenia we possess.
Because if you take a healthy volunteer and administer an amphetamine, you cause a massive dopamine release. They will develop paranoia and hallucinations, a pure positive symptom mimic.
But their cognition might actually remain sharp, and they won't necessarily look emotionally blunted.
But if you administer an NMDA receptor antagonist drugs like phencyclidine or PCP or ketamine, the clinical picture is terrifyingly complete.
An NMDA antagonist doesn't just induce hallucinations. It induces severe thought disorder, profound executive dysfunction, emotional withdrawal, and blunted affect.
It perfectly replicates the positive, negative, and cognitive symptoms simultaneously.
This implies, with tremendous force, that a failure at the NMDA glutamate receptor is fundamentally closer to the root etiology of the entire syndrome than dopamine.
So we take this powerful clinical clue back to the pathology lab. What happens to the glutamate system in the postmortem schizophrenic brain?
We find widespread, consistent evidence of hypofunction. Let's start with the AMPA receptors, which mediate fast excitatory transmission.
What's the finding there?
The transcript blueprints and the physical proteins for AMPA receptor subunits are consistently decreased, particularly in the hippocampus, but the real focus is on the NMDA receptor.
We need to explain how the NMDA receptor works because it's essentially a biological supercomputer. It's not a simple lock and key.
No, not at all. It requires glutamate to bind to it, yes. But it also requires a coagonist, glycine or D-serine, to bind simultaneously.
And even then, the channel is physically blocked by a large magnesium ion.
Exactly. The only way to pop that magnesium cork out of the channel is if the surrounding cell membrane receives enough positive electrical charge, usually provided by nearby AMPA receptors firing.
Only then does the NMDA channel open, allowing massive amounts of calcium to flood into the cell.
And that calcium influx is the trigger for long-term potentiation, the physical strengthening of the synapse. It is the molecular basis of learning and memory.
So the NMDA receptor is a coincidence detector. It only fires when multiple signals converge perfectly.
And in schizophrenia, the fundamental architecture of this coincidence detector is degraded.
The textbook details the subunits. The receptor is a tetramer made of different protein blocks.
Right, the obligatory subunit, GLOAN1, which has to be present for the receptor to function at all, is significantly downregulated across the temporal cortex, the hippocampus, and the thalamus.
Interestingly, there is a subregional upregulation of the GLOAN2B subunit in the temporal lobes.
Which, just like the CA3 dendritic spines, likely represents the cell's frantic, compensatory attempt to catch any available glutamate signal in a failing network.
But the pathology extends beyond the receptor itself. We have to look inside the cell at the postsynaptic density.
This is the physical scaffolding.
Right. If the NMDA receptor is a massive oak door, the postsynaptic density is the frame and the hinges anchoring it to the wall.
A perfect analogy. Inside the dendritic spine, there is a dense matrix of scaffolding proteins, most notably PSD95, along with neurofilament light and SAP102.
And these proteins physically anchor the NMDA receptor to the membrane and connect it to the intracellular signaling machinery.
And in the prefrontal cortex of schizophrenia subjects, transcript and protein expression for PSD95 are severely reduced.
So the hinges are rusting away. It doesn't matter how much glutamate is in the synapse. And it doesn't matter if the receptor manages to catch it, because the receptor isn't securely connected to the neuron's internal circuitry.
The signal dies at the membrane.
And this maps flawlessly back onto our macro findings in Section 1.
Because if PSD95 is degraded, the NMDA receptors collapse. Without NMDA calcium signaling, the cell cannot maintain long-term potentiation.
And if the synapse can't be maintained, the dendritic spine physically retracts.
The bridges collapse.
Yeah.
The neuropil shrinks. It is a completely unified mechanism.
But a brain composed entirely of failing excitatory glutamate signals would be pure, uncoordinated static. For information to have meaning, there must be silence between the notes.
Which brings us to the great inhibitor, gamma-aminobutyric acid, or GABA.
If glutamate is the accelerator, GABA is the brake.
But I prefer to think of GABA as the conductor of the orchestra. It doesn't just stop the music, it dictates the rhythm.
It tells the massive pyramidal excitatory neurons exactly when to fire and when to hold, sculpting raw static into coherent, oscillating rhythms.
To understand the pathology here, we have to look at the manufacturing process. The brain makes GABA directly from glutamate.
Using an enzyme called glutamic acid decarboxylase, or GAD.
Yes, it simply snaps a carboxyl group off the glutamate molecule, instantly transforming the brain's most powerful excitatory signal into its most powerful inhibitory signal.
There are two main isoforms of this enzyme, right? GAD65 and G867.
Exactly. GAD65 is concentrated at the synaptic terminals, acting as an on-demand rapid response factory for GABA release.
But GAD67 is distributed throughout the cell body, and it is responsible for maintaining the baseline resting levels of GABA, the foundational inhibitory tone of the brain.
And when we look at the GAD1 gene, which encodes the GAD67 enzyme, the postmortem data delivers one of the most highly replicated, unequivocal findings in all of psychiatric research.
What do we see?
There's a massive, consistent reduction in GAT67 mRNA levels across the prefrontal cortex.
Now, we must apply our earlier dissections. We know from the 3D stereology that the total number of neurons hasn't changed.
Therefore, this isn't a case of GABA cells dying. It is a case of the surviving cells drastically downregulating their production of the GAD67 enzyme.
The conductor is dropping their baton.
And this reduction isn't happening uniformly to every cell. It is severely concentrated in a highly specific, elite subpopulation of neurons called the parvalbumin positive, or PV positive interneurons.
The PV-positive interneurons are the fast-spiking rhythmic pacemakers of the cortex.
Structurally, they are fascinating. They send out dense, highly branched axons that form baskets wrapping tightly around the cell bodies, and specifically the axon initial segments of the massive excitatory pyramidal neurons.
So they sit right at the choke point. When the PV interneuron fires GABA, it completely shuts down the pyramidal neuron's ability to send a signal.
And because they are fast spiking, they fire in rapid, highly synchronized bursts, effectively turning vast networks of pyramidal neurons on and off simultaneously 30 to 80 times a second.
This generates what we measure on an EEG as gamma oscillations.
Gamma waves. These are the high-frequency rhythms fundamentally required for cortical binding.
When you look at an apple and simultaneously register its red color, its round shape, and its semantic meaning, it is the gamma oscillations that bind those disparate sensory inputs into a single, unified, conscious perception.
When you hold a phone number in your working memory, gamma waves sustain the circuit.
Now, let's connect the pathology. If the PV interneurons lose their GAD67 enzyme, they cannot produce sufficient GABA.
Their inhibitory signal weakens. The tight synchronous control over the pyramidal neurons degrades.
The gamma oscillations lose their sharp rhythmic timing and devolve into asynchronous noise.
The orchestra loses the beat. And clinically, this failure of PV interneuron GABAergic tone correlates directly, inextricably, with the devastating cognitive dysfunction we see in schizophrenia.
The working memory collapses because the prefrontal network literally cannot synchronize its firing patterns.
It is a stunning translation from a single enzyme transcript to a profound cognitive deficit. And the complexity deepens, because the textbook notes that this GABAergic failure does not exist in a vacuum.
Right. It is heavily modulated by the genetic architecture we touched on earlier.
Major risk genes identified in genome-wide association studies like BDNF, DIC1, and NRG1 are directly involved in the development, migration, and maintenance of these specific PV interneurons.
The genetic vulnerability strikes directly at the conductor. So we have the dopamine accelerator misfiring, the glutamate engine stalling out, and the GABA conductor losing the beat.
But a network this complex relies on secondary modulators to set the baseline acoustics of the system. We have to examine acetylcholine and adenosine.
Let's start with acetylcholine. It's essential for attention, learning, and sensory filtering.
When we examine the postmortem brains, we see a broad systemic failure of the cholinergic architecture. We measure profound decreases in the levels of nicotinic receptors, specifically the alpha-7 and alpha-4 beta-2 subtypes,
which are crucial for processing sensory information and filtering out background noise.
This perfectly explains the sensory gating deficits in schizophrenia, where patients describe feeling overwhelmed by the sensory input of a normal room.
Unable to tune out the hum of a refrigerator or the ticking of a clock.
Exactly. And alongside the nicotinic receptors, we see marked decreases in muscarinic receptors, particularly the M1 and M4 subtypes, across the hippocampus, frontal cortex, thalamus, and striatum.
Now, here is a fascinating lesson in translational medicine. Pharmaceutical companies looked at this data, specifically the alpha-7 nicotinic deficits, and saw a clear target.
They engineered highly specific positive allosteric modulators, essentially chemical dimmer switches designed to turn up the volume on whatever alpha-7 receptors were left.
In rodent models, these drugs worked beautifully, reversing cognitive deficits. But when they brought them to human clinical trials for schizophrenia, they failed spectacularly.
Why? Because the human brain is not just a larger rat brain. It is a vastly more complex, interdependent network. The textbook proposes a compelling theory for this failure.
What is it?
It suggests that in the human schizophrenic brain, the fundamental ability of the M1 muscarinic receptor to undergo allosteric modulation is intrinsically impaired.
The structural integrity of the entire cholinergic web is so degraded that pushing on a single node, the alpha-7 receptor, simply cannot overcome the systemic collapse.
It is like trying to fix a shattered transmission by putting premium petrol in the tank. You cannot target a single receptor if the network dynamics are fundamentally broken.
Which perfectly illustrates the core mandate of this deep dive. We have to think in systems, and there is no better example of a master system-level regulator than adenosine.
Adenosine is the great peacemaker of the brain. It sits at the precise physical intersection of the excitatory glutamate and the potentially psychotic dopamine systems, acting as a dual brake.
Let's trace its mechanism because it's brilliant. Presynaptically, adenosine binds to the A1 receptor, which inhibits the release of glutamate. So it cools down the excitatory engine.
Postsynaptically, it binds to the A2A receptor, which is heavily concentrated in the striatum.
Right.
And the A2A receptor forms a physical structural complex with the dopamine D2 receptor.
Right.
When adenosine binds to A2A, it allosterically decreases the binding affinity and signaling of the D2 receptor. It actively suppresses the dopamine response.
So if you have robust adenosine signaling, you keep both the noisy glutamate and the psychotic dopamine in check. But figure 12.6:1 in the textbook lays out exactly how this peacemaker fails in schizophrenia.
The transcript expression of the cellular RNA blueprints for both the canonical A1 and A2A receptors is significantly decreased.
In the dorsolateral prefrontal cortex, the A1 transcript drops, meaning the brake on glutamate is removed, contributing to the asynchronous excitatory noise.
And in the associated striatum, there is a profound reduction in both the transcript and the physical protein levels of the A2A receptor,
which removes the brake on the dopamine D2 receptor, providing a direct molecular mechanism for the hyperdopaminergia we see in living PE scans. Without A2A modulation, the striatum is hypersensitive to dopamine.
But the textbook details a phenomenal, desperate response by the living brain. While the cellular machinery is failing to produce normal amounts of A2A transcript, The actual density of the surviving A2A receptors in the striatum is upregulated by an astonishing 70%.
A 70% increase. The cell recognizes that its adenosine system is collapsing, so it frantically crams every available A2A receptor it can manufacture into the membrane.
Desperately trying to catch any stray adenosine molecule floating in the extracellular space to pull the brake on the dopamine system.
It is profound homeostatic scrambling. When we look at a post-mortem brain, we are not just looking at the primary lesion of schizophrenia. We are looking at a battlefield where the brain has spent decades engaging in massive, metabolically exhausting cellular compensation strip to save itself.
And that phrase, metabolically exhausting, provides our critical transition. Because maintaining this failing network, building new A2A receptors, trying to regrow dendritic spines in CA3, constantly trying to synchronize failing gamma rhythms, requires an astronomical amount of biological energy.
What happens if the fundamental pathology isn't in the chemical messengers at all? What if the neurons simply cannot afford the energy bill to run the machinery?
This is one of the most exciting and rapidly expanding frontiers in psychiatric neuropathology. We are descending into the mitochondrial power plants of the cell, exploring the bioenergetic dysfunction hypothesis.
The human brain is a metabolic anomaly, right? It constitutes roughly 2% of the body's total weight, yet it ruthlessly consumes 20% of the body's entire glucose supply.
It is an absolute energy hog.
Yeah.
And for a cortical neuron to function, that glucose has to cross the blood-brain barrier and enter the cell.
That entry is controlled by highly specific doorways, the glucose transporters.
The most critical for our discussion are GLUT1, which sits on the blood-brain barrier and the astrocytes, and GLUT3, which is the primary doorway into the neurons themselves.
And the post-mortem transcriptomic data highlights a significant decrease in the transcripts for both GLUT1 and GLUT3, specifically in the pyramidal neurons of the dorsolateral prefrontal cortex.
Right at the absolute beginning of the supply chain, the doors are closing. The prefrontal cortex is being starved of its primary fuel.
But it gets worse, because even the glucose that manages to get inside the cell is mishandled.
Once glucose enters the cytoplasm, the first step in extracting energy is glycolysis. This begins with the enzyme hexokinase, which phosphorylates the glucose, trapping it inside the cell.
Hexokinase can float freely in the cytosol, but for maximum efficiency, it physically binds to the outer membrane of the mitochondria.
This couples the initial breakdown of glucose directly to the mitochondrial furnace.
It's an assembly line. But in the schizophrenic prefrontal cortex, the ratio of cytosolic to mitochondrial hexokinase is significantly increased.
The assembly line is uncoupling. The hexokinase is detaching from the mitochondria, rendering the energy extraction highly inefficient. Furthermore, the overall transcript expression of the key rate-limiting glycolytic enzymes, including hexokinase and phosphofructokinase, is decreased, and the actual enzymatic activity of hexokinase drops.
The entire glycolytic capacity of the prefrontal cortex is shrinking. And critically, researchers have mapped the genes encoding these glycolytic enzymes, and they fall directly into the genetic risk loci identified in schizophrenia genome-wide association studies.
This isn't just a side effect of poor diet or antipsychotic medication. The genetic blueprint for the metabolic power grid is faulty from conception.
But the most staggering bioenergetic anomaly, the finding that truly illustrates a system in crisis, is found in the next metabolic stage, the tricarboxylic acid cycle, or TCA cycle.
The Krebs cycle. Every medical student memorizes this and usually hates it.
It is a continuous looping biochemical pathway inside the mitochondria. Metabolites enter, they are systematically oxidized by a series of enzymes, they produce electron carriers like NADH to drive ATP production, and the cycle repeats.
Because it is a continuous loop, logic dictates that the whole cycle should speed up or slow down as a single unit.
But transcriptomic and proteomic studies of the dorsolateral prefrontal cortex in schizophrenia reveal a bizarre, highly specific fracturing of the cycle.
When researchers measure the enzymatic activity, they find that the enzymes driving the first half of the TCA cycle, specifically aconitase, which converts citrate to oxocitrate, and alpha-ketoglutarate dehydrogenase are significantly downregulated. Their activity is suppressed.
But when they look at the enzymes in the second half of the cycle,
Enzymes like succinate dehydrogenase and malate dehydrogenase, their expression and activity are heavily increased.
we have to translate what a fractured cycle means for the living cell. If the first half is slowing down and the second half is working in overdrive, you have a massive biochemical bottleneck. The primary flow of carbon from glucose is blocked.
It indicates profound cellular starvation. Because the primary route is blocked, the neuron is frantically trying to generate ATP by shunting alternative metabolic fuels directly into the second half of the cycle.
It begins engaging in anaplerosis, pulling in amino acids like glutamine or metabolizing local lipids just to keep the succinate dehydrogenase pumping to keep the lights on.
The cell is essentially cannibalizing its reserve fuels to survive a fundamental blockade in glucose metabolism.
And we see the consequences of this distress cascade all the way down into oxidative phosphorylation, the final stage of energy production via the electron transport chain embedded in the inner mitochondrial membrane.
The postmortem data here is robust. There are decreased transcript levels for critical ETC complexes and the ATP synthase machinery across the prefrontal cortex.
Advanced proteomic analyses utilizing mass spectrometry have physically measured significant decreases in complex 1 and complex 3.
And we see these ETC perturbations not just in the prefrontal cortex, but in the temporal cortices and the basal ganglia. The power grid failure is systemic.
Now, to be thorough, the textbook notes that when researchers actually put these mitochondria under electron microscopes to look at their physical morphology, the results are hotly debated.
Some studies report that the mitochondria are smaller or fewer in number. Others look at different cell lines and claim the physical structure is perfectly intact.
Morphological postmortem studies of mitochondria are notoriously difficult due to rapid tissue degradation after death, right?
Exactly. But the biochemical consensus is undeniable. Regardless of how they look, their functional bioenergetic capacity is severely compromised.
And this metabolic failure circles us directly back to the very first macroscopic finding we discussed, the reduced neuropil in the prefrontal cortex.
Because which specific layers of the cortex have the absolute highest baseline metabolic demand?
Layers 3 and 5. The deep projection neurons. They maintain massive, sprawling dendritic arbors and send axons across vast anatomical distances. They are the energy hogs of the energy hog.
If the prefrontal cortex is experiencing a TCA cycle bottleneck and a failure of the electron transport chain, layer 3 is the first to starve.
It simply cannot afford the ATP required to maintain its millions of dendritic spines, nor can it afford the energy to power the NMDA receptors or synthesize GAD67.
The bridges collapse because the city cannot pay the power bill to maintain them. It all connects.
A completely unified mechanistic framework. But a profound question remains for our detective. We have identified the missing bridges, the chemical imbalances, and the failing power grid. But when exactly did the house fall apart?
Schizophrenia rarely manifests as full-blown psychosis before late adolescence or early adulthood.
Yet we know the pathology doesn't just spontaneously generate on a patient's 20th birthday. To find the origin, we have to look backwards in time. We enter the realm of neurodevelopment and the environment.
Schizophrenia is, at its absolute core, a neurodevelopmental disorder. The brain doesn't break, it grows into its own pathology. And to understand that growth, we must examine the intersection of the hardware and the software, genetics and epigenetics.
Let's begin with the hardware. The genetic vulnerability is undeniable. Schizophrenia demonstrates a heritability estimate of roughly 79%.
But the clinical translation of that number requires nuance. If you look at monozygotic twins, identical twins who share 100% of their DNA sequence, the concordance rate for schizophrenia is roughly 33 to 50%.
If one twin develops the illness, the other twin has a high risk, but it is unequivocally not a certainty.
Which proves a fundamental biological axiom. DNA is not destiny. The genetic blueprint provides the vulnerability, but external environmental factors are absolutely required to trigger the manifestation of the disease.
Furthermore, schizophrenia is highly polygenic. It is not caused by a single broken gene like Huntington's disease.
No, it is the cumulative result of thousands of common alleles, each contributing a microscopic sliver of risk, sometimes compounded by rare, highly penetrant copy number variations.
The textbook highlights a few major genetic hitters found via GWAS that directly link back to the mechanisms we've discussed. Genes like NRG1, which guides neuronal migration and myelination, DISC1, which acts as a massive scaffolding hub for synaptic proteins, and ZNF-ET4A, which regulates gene transcription.
The hardware provides a vulnerable, easily disrupted foundation. But the software running on that hardware is what ultimately dictates the outcome. That brings us to epigenetics.
Epigenetics is the physical molecular bridge between the environment and the genome.
Every single cell in your brain contains the exact same sequence of DNA. But a parvul-Bowman interneuron looks and behaves entirely differently from a layer 3 pyramidal cell because it is expressing a different subset of those genes.
Epigenetics is the system that turns specific genes on or off.
The most prominent mechanism of epigenetic control is DNA methylation. The cell attaches a tiny chemical tag, a methyl group, directly onto the cytosine bases of the DNA strand.
To visualize this, think of the DNA as a long string of thread wrapped tightly around protein spools called histones. If the thread is wrapped loosely, the cellular machinery can read the code and build the protein.
But when the DNA is heavily methylated, it triggers chromatin remodeling. The thread is pulled incredibly tight around the spool. The gene is physically locked away, silenced, unable to be transcribed.
And this epigenetic silencing is actively managed by enzymes called DNA methyltransferases, or DNMTs.
In the postmortem brains of schizophrenia subjects, researchers made a staggering discovery regarding these enzymes. They found that DNMT1 and DNMT3A are highly overexpressed, specifically within the cortical GABAergic interneurons.
The exact same parvalbumin interneurons where we saw the catastrophic loss of the GAD67 enzyme.
Exactly. The overactive methyltransferases are hypermethylating the promoter region of the GAD1 gene, pulling the thread tight and physically preventing the cell from manufacturing the GAD67 enzyme.
The environment isn't mutating the gene, it is burying it alive.
And because the epigenome is highly dynamic, it's exquisitely sensitive to environmental insults, particularly during the prenatal period.
The textbook highlights epidemiological data showing that adverse intrauterine conditions, maternal malnutrition, such as the famous Dutch hunger winter cohorts, or severe maternal viral infections, significantly increase the risk of the offspring developing schizophrenia decades later.
During a severe maternal infection, pro-inflammatory cytokines can cross the placenta and enter the fetal brain, triggering an epigenetic cascade.
The trauma of the infection forces the developing fetal brain to hypermethylate critical neurodevelopmental genes, setting the brain on a subtle detour.
The hardware is intact, but the software has been rewritten by the environment before the child takes their first breath.
So we have a vulnerable blueprint and an environmental insult rewriting the software in utero. Now we have to trace the timeline of this developmental detour. The brain is built in distinct, overlapping stages.
The first major stage is neuronal migration, peaking in the second trimester. Neurons are born in the ventricular zone, deep in the center of the brain, and they must physically climb a scaffolding of radial glia to reach their precise designated layer in the outer cortex.
Now, if you read older literature, there was a very popular theory of migration failure in schizophrenia. Researchers claimed to find ectopic neuron cells that had gotten lost on their climb and were stuck in the white matter underlying the prefrontal and entorhinal cortices.
It was a compelling narrative that perfectly supported the neurodevelopmental model. However, scientific rigor requires us to update our models.
What did modern follow-up studies find?
When modern neuropathologists conducted deeply rigorous follow-up studies, carefully controlling for the natural, highly heterogeneous distribution of interstitial neurons in the normal human brain, they could not replicate the findings of massive ectopic clusters.
The consensus has shifted. While subtle micro-migration errors may occur, gross, widespread neuronal migration failure is likely not the primary driver of schizophrenia.
If the cells reach the correct destinations, the focus shifts to the next stage, spinogenesis and pruning.
The building of the bridges and the selective destruction of the weak ones.
A human brain reaches its absolute maximum synaptic density between 2 and 4 years of age. A toddler has roughly double the number of synaptic connections as a healthy adult.
Because the brain essentially builds an over-connected, chaotic web and then relies on experience to act as a gardener.
Over the next decade, and accelerating massively during adolescence, the brain aggressively prunes away the weak, unused synapses to streamline the circuits and increase processing efficiency.
This physiological pruning leads us directly to one of the most dominant contemporary theories of schizophrenia, the excessive pruning hypothesis.
The core concept is that during the critical window of adolescence, the cellular gardener becomes hyperactive, pathologically overzealous, and begins severing vital, necessary synaptic connections.
And what drives the gardener? Recent landmark genetic studies have pointed the finger directly at the immune system, specifically the complement cascade, like the C4A gene.
The immune system is basically tagging healthy synapses with a molecular eat-me signal, and the brain's microglia happily comply, devouring the connections.
Which brings our entire conceptual model full circle. The excessive adolescent pruning is the exact mechanism that generates the reduced neuropil, the missing dendritic spines in layer 3 of the prefrontal cortex, and the collapse of the glutamate NMDA architecture we discussed at the very beginning of the deep dive.
It is not an adult neurodegenerative disease. It is an adolescent neurodevelopmental derailment.
And as the bridges are being aggressively pruned, the surviving neural highways are undergoing myelination. Oligodendrocytes wrap the long axons in a fatty myelin sheath, massively increasing the speed of electrical transmission.
Myelination is a remarkably protracted process. In the prefrontal cortex, it continues actively well into a person's late 20s.
Which means there is a massive 2-decade-long window of vulnerability where environmental exposures can disrupt the final maturation of the brain's highest order networks.
It perfectly explains why heavy cannabis use during the critical mid-adolescent window is such a potent environmental risk factor for triggering psychosis in genetically vulnerable individuals.
You are introducing a powerful exogenous neuromodulator precisely when the prefrontal cortex is undergoing its most delicate pruning and myelination phase.
And we can actually observe the subtle physical manifestations of these developmental detours long before the onset of hallucinations or delusions.
Clinicians often observe neurologic soft signs, or NSS, in children who later develop schizophrenia.
These are subtle deficits in sensory integration, motor coordination, or the retention of primitive reflexes. A slight clumsiness, an inability to perfectly mirror a complex hand movement.
They are not specific diagnostic markers for schizophrenia, but they serve as a visible clinical readout of the underlying genetic and epigenetic turbulence. They confirm that the neurodevelopmental trajectory has been subtly off-kilter for years.
We have built a massive, intricate architecture today.
We are entering our final section, integration and clinical reasoning. It is time for Medlock Holmes to put all his clues on the board and synthesize a cohesive, actionable picture.
But before we build the final clinical formulation, we have to acknowledge the elephant in the modern psychiatric room, the inflammation hypothesis.
You cannot read a contemporary psychiatric journal without confronting the immune system. The brain is not immune privileged in the way we once thought.
It has its own resident immune cells, the microglia, and we already noted that macaque monkeys exposed to antipsychotics showed increased glial densities.
Furthermore, genome-wide association studies show that the most significant genetic risk loci for schizophrenia reside directly within the major histocompatibility complex, the master regulator of the immune system.
We know that acute psychosis is often accompanied by spikes in pro-inflammatory cytokines circulating in the blood and cerebrospinal fluid.
And in certain animal models of maternal immune activation, administering anti-inflammatory agents can actually rescue some of the cognitive and behavioral deficits. It paints a picture of schizophrenia as a chronic, low-grade neuroinflammatory disease.
But I have to push back here because Chapter 097 explicitly warns us that the data is not a clean, straight line.
Right. The chapter highlights a massive 2019 epidemiological study that measured C-reactive protein, or CRP, the gold standard biomarker for systemic inflammation.
The study paradoxically found that elevated CRP might actually exert a protective effect against developing schizophrenia.
How do we reconcile that? If inflammation is the axe destroying the synapses, how can a marker of inflammation be protective?
It is the ultimate paradox of neuroimmunology, and it proves that inflammation equals disease is far too simplistic a model.
The key is likely timing and context.
Exactly. A massive inflammatory spike in utero might cause disastrous epigenetic hypermethylation, but an acute inflammatory response in a young adult might represent a necessary protective physiological process. Perhaps the immune system actively trying to clear out toxic metabolic byproducts from the failing TCA cycle.
The immune system is intimately involved, but it is a double-edged sword. We are still mapping which edge is cutting the pathology and which edge is trying to heal it.
Which is a perfect summation of the entire illness. So let's step back and synthesize the mental model for the clinician.
We have journeyed from macroscopic volume loss down to the methylation of individual cytosine bases. How does a modern psychiatrist utilize this data in practice?
By fundamentally reframing their definition of the illness. Schizophrenia is not a focal lesion. It is a disease of abnormal connectivity. It is a failure of temporal and spatial integration at the molecular, synaptic, and circuit levels.
Mechanism to manifestation. Let's trace it one last time. Imagine you are a registrar in the clinic. You are sitting across from a patient who is exhibiting profound negative and cognitive symptoms.
They are emotionally withdrawn, their speech is impoverished, they cannot hold a train of thought to answer a simple question.
As a clinician, you should not just be checking off symptom boxes. You should be actively conceptualizing the biology.
You should visualize the dorsolateral prefrontal cortex.
Yeah.
You picture the excessive adolescent pruning that stripped away the dendritic spines on the layer 3 pyramidal neurons.
Because those physical bridges are missing, the excitatory NMDA receptors are unanchored and failing.
This glutamate failure cascades down to the parvalbumin interneurons, which, burdened by epigenetic silencing, fail to produce the GAD67 conductor.
The gamma oscillations collapse into static, destroying working memory.
And underneath it all, the mitochondrial TCA cycle is bottlenecked, starving the cortex of the ATP it desperately needs to rebuild.
It is a profound failure of the prefrontal executive network. And what about when that same patient returns months later in the grip of acute positive symptoms?
They are paranoid, experiencing auditory hallucinations, attaching terrifying meaning to random events.
Then your conceptualization shifts to the subcortical regions and the associative striatum. The failing prefrontal cortex loses its top-down regulatory control.
Simultaneously, the adenosine A1 and A2A modulatory systems collapse. Without these breaks, the subcortical dopamine engine redlines.
Nascent dopamine synthesis skyrockets, flooding the synapses, inappropriately tagging internal thoughts and external stimuli with massive aberrant salience, generating the hallucination.
It is a beautifully, tragically interconnected system. You cannot tweak one dial without affecting the others.
Dopamine drives glutamate, glutamate drives GABA, adenosine modulates both, and the geneticical genetic software dictates the structural integrity of the entire web.
Which is precisely why the future of psychiatry is moving away from broad, monolithic diagnoses. The DSM criteria of schizophrenia will eventually be replaced by neurobiological biotypes, identified via next-generation sequencing and transcriptomics.
Patient A might present with psychosis driven primarily by a complement-mediated pruning defect, while patient B's psychosis is driven by a profound bioenergetic hexokinase failure.
Understanding the specific network failure will allow for precision, system-level engineering, rather than blunt pharmacological hammers.
The ultimate goal of this deep dive was to build that dynamic, systems-level mental model. But before we close, Chapter 097 demands we test it.
We aren't just here to muse philosophically.
No, this is for the clinicians listening, the registrars prepping for their board exams. I am going to present 5 clinical and mechanistic scenarios, a rapid-fire, single best answer format. I will lay out the question. We will give you a brief beat to lock in your clinical reasoning, and then we will unpack the precise rationale. Are you ready?
Let's put our listeners in the hot seat.
Scenario 1. A concept question. A researcher uses unbiased 3D optical fractionator stereology to examine the dorsolateral prefrontal cortex of a deceased patient with chronic schizophrenia. Which of the following best describes the most robust, consistently replicated finding regarding the neurons in this region? Take a moment to think.
The answer is an increased neuronal density due to a reduced neuropil volume, without any decrease in the absolute neuronal number. Remember our opening metaphor. The rooms of the house are still there, the cell bodies survive. But because the hallways and wiring, the dendritic spines and axonal arbors have been excessively pruned away, the entire structure condenses, packing the surviving cells tighter together.
Spot on. Scenario 2. A mechanism question. You are evaluating a patient exhibiting profound deficits in working memory and executive function. Based on postmortem molecular pathology, profound dysfunction in which of the following specific interneuron subtypes is most strongly correlated with these cognitive deficits? Take a moment.
The answer is the part-of-human-positive interneurons associated with reduced GAD67 expression. These are the fast-spiking conductors. Without GAD67, they cannot produce the basal GABAergic tone required to generate the high-frequency gamma oscillations that physically bind cognitive processes together in the prefrontal cortex.
Brilliant. Scenario 3. Clinical reasoning. A 20-year-old patient presents to the emergency department with a first episode of acute psychosis. A structural CT scan reveals significant enlargement of the third and lateral ventricles. As the attending psychiatrist, how should you interpret this specific radiological finding? Lock in your answer.
You must interpret that structural brain changes are present at the onset of the illness, but crucially, the finding lacks diagnostic specificity for schizophrenia. Enlarged ventricles represent exacuopassive expansion and are seen in bipolar disorder, normal aging, and even in the perfectly healthy relatives of the patient who share their genetic risk alleles. It is a marker of vulnerability, not a definitive diagnosis.
Absolutely. Scenario 4. Mechanism. Transcriptomic and proteomic analyses are performed on the mitochondria within the dorsolateral prefrontal cortex of a schizophrenia subject. Which specific bioenergetic abnormality has been consistently identified within the tricarboxylic acid or TCA cycle? Think about it.
The answer is a paradoxical bottleneck, a decrease in the activity of enzymes in the first half of the cycle, such as aconitase, coupled with an increase in the activity of enzymes in the second half, such as succinate dehydrogenase. This biochemical traffic jam indicates profound cellular starvation, forcing the neuron to shunt alternative metabolic fuels like amino acids into the cycle just to maintain baseline survival.
Final scenario, scenario 5. Concept. The widely accepted excessive pruning hypothesis posits that aberrant synaptic elimination directly contributes to the pathophysiology of schizophrenia, during which specific human developmental period is this normative synaptic pruning process most aggressively active in the cortex. Take a moment.
The answer is adolescence. While peak synaptic density is achieved in early childhood, the aggressive, complement-mediated clearing of those synapses accelerates massively during the adolescent years. This provides the exact temporal window for the pathology to develop, perfectly coinciding with the typical prodromal and first episode clinical presentation.
Exceptional. If you found yourself struggling with any of those scenarios, we highly direct you back to the source text, chapter 097 of Kaplan and Sadok's comprehensive textbook of psychiatry, to solidify the neurobiology.
We have covered a breathtaking amount of ground today.
We really have. We journeyed from the macroscopic weight of the brain down to the subtle methylation of individual cytosine bases on the DNA strand. We mapped the failing bridges, the struggling power grids, and the chaotic chemical messengers. But as we wrap up this deep dive, I want to leave you, the listener, with a final, lingering thought to mull over on your commute or in the clinic.
A provocative thought to end on.
We discussed how the epigenome is the physical canvas where the environment violently paints over our DNA. And we discussed how synaptic pruning physically reshapes the architecture of our mind well into our late 20s. So, if the genetic risk alleles are present at the exact moment of conception, but the environmental viral trigger happens in utero, and the massive complement-mediated synaptic pruning doesn't tear the bridges down until adolescence, at what exact definitive moment does schizophrenia actually begin?
It is the ultimate philosophical and biological question of neurodevelopment.
Is the fate of that brain written inextricably in its genes before the child is even born, or is it a precipitous tipping point? A threshold reached only after 1,000 microscopic cellular compensations, the frantic upregulation of adenosine A2A receptors, the desperate shunting of amino acids into the TCA cycle, the hyperactive sprouting of spines in the CA3 hippocampus finally fail under the crushing energetic load of entering young adulthood.
It forces you to think about the immense lifelong plasticity of the human brain.
Yes, that plasticity is the source of the vulnerability. It is what allows the cellular gardener to prune away far too many branches, leaving the house disconnected. But perhaps, eventually, as we learn to map these biotypes and truly understand these interconnected compensatory networks, that exact same lifelong plasticity will be the master key to reversing the condition entirely.
It's a hopeful thought.
Thank you for joining us on this deep dive. Keep questioning the obvious, keep looking for the missing bridges in the dark, and we will see you next time.